Hi, my name is Jamie Robinson. Today I'll be talking about work that I and my research group did investigating pressure development mechanisms in sugar maple. So, as a bit of background, sugar maples will develop elevated stem pressures when ambient temperatures regularly fluctuate above and below 0 degrees C. That is, the tree regularly freezes and then thaws over. Now, because of this elevated stem pressure, you can relatively easily harvest large amounts of sap by simply drilling what's known as a tap hole straight into the stem. And in the image there you can see an example of a commercial setup where multiple trees have been tapped, hooked up through a piping network that drains to a central collection location, often with a degree of vacuum applied to the system to improve on yields. So, the pressure that develops within sugar maple develops within a region of the stem known as a xylem. In the image there you can see a sugar maple sapling, where in the centre you have a region known as the pith, and then you have the outer layers of the cambium or the bark, and between them you have a region known as the xylem, which is the region through which sap flows up from the roots through the stem. If we look at the xylem up close, we can see that it's composed of many individual cells all packed closely together. The ones most of interest for pressure development are the vessels. These are large diameter sort of tubes that run vertically through the xylem, and it's through these tubes that sap flows up the tree. And you can also see the fibres. These are close-ended, short, dead cells that are, in sugar maple at least, primarily believed to be filled with gas, and in most tree species serve a structural support role. Now, in sugar maple, the pressure that develops within the stem is believed to develop due to the compression of this gas in the fibres. Specifically, as a tree freezes over, the temperature of this fibre gas drops, the pressure drops, and this leads to water being drawn into the fibres where it freezes, sort of being drawn up from the roots and through the vessels. During a subsequent thaw, this temperature in the fibre increases and the pressure increases as the ice melts, and this results in a net increase in stem pressure that builds over multiple cycles of freezing and thawing. Now, this model was first proposed about 40 years ago, and while there has been some follow-up and some additions made over the years, the overall body of literature on sugar maples is fairly limited. Now, my role within this research group has been to try and improve upon this limited body of research. Specifically, I was using different imaging techniques to examine the anatomy of sugar maple and to investigate this model of pressure development to try and determine whether experimental evidence supported the hypothesized mechanism. Now, some of the earliest work on this project in this vein was done by a former PhD student named Tanaya Driller, who since graduated. Now, she looked at cut pieces of maple stem and imaged them using light optical microscopy and scanning electron microscopy. Light optical microscopy uses lenses to focus light to get high magnification, while scanning electron microscopy uses an electron beam to scan across a surface to generate a high-resolution image. Now, by taking these images of cut pieces of maple stem, she could examine the anatomy of the xylem, looking at the distribution of the various cells as well as their sizes, and using particularly high resolution, she could make out small pores that are present that interconnect various cells within the xylem. Now, in addition to those sort of flat cuts, she also made what are known as macerations, where you break down a sample of maple stem in order to separate out the individual cells, which can make imaging some of the various different cell types more easy. Now, well, this is some of the work that she was involved in, and she was also involved in some cryo-SEM, where we were trying to use liquid nitrogen to rapidly freeze stems to fix the moisture in place before imaging. The work that I'm primarily going to discuss, which is the work that I was involved with, was the microcomputed tomography. Now, microcomputed tomography is an imaging technique whereby you shine X-rays on an object and capture a flat image. Here you can see a flat image of a piece of maple stem. This is equivalent to what you would get if you take an X-ray of someone's arm to see the bones, for example. However, unlike just imaging an arm, here we take a series of X-rays at different angles, and using those X-rays we can reconstruct a full 3D image of an object. This is very useful for letting us see the full internal and external structure without requiring us to cut into the maple stem to take the image. Now, the micro-CT work that was done was primarily done at a research facility known as the Australian Synchrotron, which is based in Melbourne, Australia. Now, this is a large-scale building that primarily houses a huge metal ring that holds accelerated electrons. Now those electrons can be used to generate a variety of different outputs for various research stations stationed around the ring that use them for different imaging applications. For my work, there were 2 stations that used it for micro-CT work. These stations allowed us to generate high-resolution micro-CT scans in relatively short spaces of time, often with larger samples than would be reasonably possible with some of the benchtop setups that do exist. Now, the first body of work that I'm going to discuss was done at the MCT beamline. This is one of the stations at the Australian synchrotron that's particularly tuned for high-resolution work of somewhat smaller samples. You can see in the image down here an example of our imaging setup. Just off screen is the source of the X-rays, effectively a shutter that opens to let them in. And on the other side you have our camera that detects the X-rays to get our image. And in between them you have a piece of maple stem that we're actually imaging. In this case, because of the size of the facilities and the somewhat small field of view of the imaging apparatus, we could only image somewhat small sections of maple stem. This is all mounted on a turntable so we can rotate that sample to take the series of projections necessary to generate our 3D image, and we actually only image a very small part of that overall stem. So here you can see an example of one of those 3D images of just a small part of that larger stem. Here, what's notable is that the resolution is high enough that we can resolve the individual cells within the xylem. If you look at the zoomed-in image, you can see the fibers in the vessels, with many of them appearing very dark. With contrast in these images being based on density differences, the darker cells are those that are filled with gas. And we were using this approach to be able to observe which fibers were filled with gas so that we could evaluate this. For this work, we were trying to examine the effects of sucrose on this gas content within the fibers. One of the additions that's been made to the basic theory of pressure development is that the high sugar content of maple sap, which is primarily sucrose, plays a stabilizing role, preventing that gas in the fibers from dissolving over time under ambient conditions. To test this, we use this custom rig here. This is effectively just a pressure vessel which is pressurized with nitrogen. It contained either a solution of 2% sucrose or just water, and we forced this solution through stem segments that were mounted down the bottom, effectively flushing the vessel content so we could observe the effects of maintaining or removing the sugar within the sap in the vessels. Just as an example of what some of those results looked like with us imaging several maple stems, here you can see a sample that was imaged initially with most of the fibers being gas-filled. And after the perfusion with water over 2 hours, you can see that most of the fibers appear much lighter in colour, that is, they're no longer gas-filled, they're filled with water. By contrast, here's another stem segment, and here's that same stem segment after being perfused for 2 hours with sucrose solution, and here most of the fibers still appear to be gas-filled. So using micro-CT, we were able to obtain direct evidence in support of the hypothesis that the sucrose is playing a stabilizing role in that fiber gas content. Now, the other big body of research that I was involved with was the freeze-thaw work. As I've talked about earlier, sugar maples develop elevated stem pressures when they undergo periods of freezing and thawing. We wanted to replicate this setup experimentally, imaging a maple sapling across a freeze-thaw cycle to see if we could try to capture that change in gas content in the fibers. Now, this was done on the IMBL, which is another station at the Australian synchrotron. This is tuned to slightly lower resolutions but has a much larger space to mount samples. Down the bottom there you can see the experimental setup that we used. You can see our sapling here. This is mounted inside a polystyrene chamber to insulate it from the surrounding environment. On one side is our detector to take our image, and our other side is our X-ray source. And we image a small part of that overall sapling that falls within the range of the X-ray beam. Now this is all mounted on a turntable to rotate that whole rig to take our series of projections to reconstruct our 3D image. Now this rig was, as I said, run in 2022. This is an improvement upon an earlier rig that was run by the PhD student I talked about earlier. That earlier rig used dry ice to rapidly freeze maple saplings and then imaged them as they slowly unthawed. For this rig we used a gas tube. This contained nitrogen gas that was passed through a container of liquid nitrogen to rapidly cool it before it entered the chamber. By controlling the flow rate of gas into the main chamber, we were trying to control the temperature to recreate more realistic freeze-thaw conditions, like what a sapling or a full tree would experience in nature. Now, while this rig did work somewhat, it was not super stable on the temperature control and it wasn't super reliable. In particular, we kept emptying out nitrogen gas cylinders in the middle of our runs. So, because of this, we chose to redesign the rig and then run further experiments in late 2023. In the images there, you can see an example of our new rig, both on site here at the University of Canterbury and on the actual experiments and at the IMBL. Now, unlike the previous rig, which used cooled nitrogen gas, this rig used a very different system. If we look at this cross-section of the base, you can see that within the base is mounted a radiator, a fan, and a small heater. There's actually 3 of these mounted around the base. With this design, the fan draws air from the main chamber through the radiators, up the double-walled construction, and then back into the main chamber. Now, the radiator is fed by a mixture of ethylene glycol and water at about negative 17 or 18 degrees or so, and by controlling the flow rate of this mixture through the radiators, the fan speed, and then the heater, we could achieve somewhat stable temperature control over a range of 7 to minus 7 degrees, and thus recreate more stable freeze-thaw conditions like what would occur in nature. So, we ran several trees through these simulated freeze-thaw cycles, imaging them across the process. Here you can see single slices of a 3D image for a single tree, from an image taken before the tree was frozen, from an image taken while it was frozen, and from an image taken after it was thawed. Now, unlike with the work done at the MCT beamline, the resolution of this setup is not sufficient to resolve the individual cells that compose the xylem. However, we do see distinct changes in the xylem across the freeze-thaw process. Particularly if you can see in this region, the overall region of the xylem appears much lighter, whereas after thawing it mostly returns to its initial state. Now, because the contrast in these images is again based on density differences, we believe these changes correspond to a decrease and then an increase in fiber gas content, with water freezing in the fibers resulting in an increase in the overall density of that region. So while we can't resolve the individual fibers, the evidence that we've obtained here, which was consistent across multiple saplings, appears to be consistent with the hypothesized mechanism for pressure development in sugar maple. So, to summarize, through the various experiments we've performed, we've obtained detailed measurements of maple anatomy that weren't available previously. We've obtained strong evidence in support of the stabilizing role of sucrose on that fiber gas content, and that work has actually recently been published in the Journal of Tree Physiology. And finally, we've obtained evidence for this hypothesized freeze-thaw mechanism in fiber ice formation occurring within the stem of living maple saplings. And this is particularly notable as the evidence that was used to establish the theory was based on more indirect measurements using cut pieces of maple stem. So observing this in an actual living sapling is quite significant. So that summarizes the work we've done, and thank you for listening.